And so we're trying to stay connected on what we're doing and make crowdsourcing pathologists kind of something that can happen often and support all the different artificial intelligence development and validation. So I think that's really the kind of space we're working in to create the data that these algorithms need and the data that we need to validate these algorithms. So with that, Amgad, I'm going to turn it over to you to maybe, I don't have your full bio or anything, but I know you're from Emory University. If you could just say a few words about where you are in your career and the projects you're working on. Thank you for being on here today.
Thank you so much, Brandon and Weiji, for the wonderful, wonderful introduction. And it's always a pleasure to connect with you and collaborate. So my name is Mohamed Amgad. I am a visiting predoctoral fellow at Northwestern University. That's where I'm fully stationed at the moment in Chicago. I am finishing my PhD at Emory University in tandem with my predoctoral appointment. I finished my medical training originally in Egypt from Cairo University. That's where I got my medical degree before moving to the US for my PhD in biomedical informatics under supervision of Dr. Lee Cooper, who is now heading the computational pathology effort at Northwestern. So it's wonderful to be here. Thank you everyone who is joining us today. And you know, I really look forward to sharing our work with you on crowdsourcing, basically engaging a very wide audience in the generation of ground truth data or real reality data, quote unquote, to basically compare pathology images and to train models. This follows from previous work we've done, and I'm really happy to share this work with you. This is my personal website here, and we also have a website for the dataset that I'm going to be sharing later on. Regarding the questions, feel free to, if you have a very burning question, feel free to ask in the middle, though I will be giving space for questions every few slides, every 4 slides or so between the sections, where there will be just space for questions related to these particular subsections. So thanks again for joining, and let's get started. So first of all, I'm going to start with a simple question. Why should we bother with crowdsourcing the annotation of medical images? Why is that an important problem? And this really stems from a general issue, or the broad scheme of issues that we face in the now somewhat mature field of computational pathology. So computational pathology is this field where we have, you know, normal pathology, or in the field of pathology is where you have a pathologist, a medical doctor who is trained to look at these slides of mostly cancer, most often, and they look at it under the microscope and they try to determine what is the grade of the tumor, basically how ugly do the nuclei look, how infiltrative they are, what is the likelihood that this patient will survive to a certain, you know, what's the survival prospects of people with this kind of microscopic picture, and so on. So this is pathology, and computational pathology is the field where we try to really quantify all of these things in a reproducible and very scalable way, sometimes in a way that even is not possible using manual methods. So computational pathology really starts with the technology of whole slide scanning. And what you're seeing here is just a small part of a whole slide image. Basically, you take these slides and you put them under a scanner and you scan them at an extremely high resolution, usually, you know, gigabytes or gigapixels, sorry, in size, and actually gigabytes in actual size, disk space. So it's an extreme scale kind of problem, and I sometimes when I talk with my friends, they become surprised when I tell them that we share a lot with the things like Google Earth and things that deal with extreme scale at the level satellite images, and so on. And it's because we deal with similar problems in terms of scale. Now, in the specific problems that I work on, we try to extract what we call semantic representations or concepts from these slide images. So this is what you're seeing here is a snapshot from a breast cancer patient from the public data set called TCGA. And you can see here that there is a lot of variability in the elements. And let me maybe show this figure here, where you can see that, you know, within the same tumor, there's a lot of intratumoral heterogeneity. The tumor is seen here in the upper left corner, and then there are things that happen within the tumor itself, including lots of characteristics that are specific to tumor nuclei and things that are ugly or infiltrative. There's necrotic debris, which is basically dead tissue because the tumor is outgrowing its blood supply. And there are the famous TILs, or tumor infiltrating lymphocytes, that would sometimes be at the tumor margin, sometimes would actually go inside the tumor to try to fight back the cancer. So these are your immune system. And there's all sorts of you know, things, normal glands, normal epithelia, immature fibroblasts, a lot of things. So the challenges of computational pathology, beside the huge scale, you know, with over 10,000 regions per slide, 500 nuclei per slide. By the way, regions is kind of these low resolution things that you see, these big blocks of stuff, whereas nuclei are these small individual cells. These are the things that create, that are made of DNA, basically, that encode everything that we have. And other problems that we have in computational pathology is the variability. There's a lot of differences between the various labs, analytical and analytical factors related to the scanning, different scanning manufacturers and staining protocols, and so on. And the specific problem that I'm really trying to address here is that people talk a lot about big data. It's all over the news, but really there's big unlabeled data in the medical domain, and especially in pathology, but there's very small labeled data. We don't actually have what we call ground truth, basically labels for these individual nuclei. There's very little data out there where the experts, namely the pathologists, say that this particular nucleus is a lymphocyte and belongs to our immune system, or this particular nucleus is mitotic and is dividing. There's very little of this data out there, and that has been kind of a key bottleneck in hindering the advancement of this field. Another thing that we kind of face, another kind of challenge in computational pathology, is how critical the application is. With many of the other applications, maybe not self-driving cars, because that's maybe a critical application, but generally speaking, a lot of the other problems that are solved by AI, let's say game playing or things like that, the application is not so critical. Whereas in the medical domain, especially if you're making a diagnosis in pathology, where treatment decisions will ensue, and even if the model is not making the decision, and even if it's just biasing the pathologist, that's still a critical application that could result in life and death. And this is where the domain of interpretability becomes important. In other words, being able to understand why our deep learning models, or machine learning models in general, are making the decisions they do, and when they fail, we understand why they fail. So this is where interpretability is important. And there's basically two competing paradigms that are currently and I'm not arguing for one over the other. Both of them are important. But the one that I'm involved in is the detection of these individual regions and nuclei, and then using them for various things, for survival prediction, or for prediction of response of the immunotherapy response, and so on. This is called a concept bottleneck model. Let me just jump to this area here, this part here. So this is called concept bottleneck, which is the idea that instead of making the prediction going from this is a radiology image and there is this black box model that I don't know exactly what it's doing, but it's very accurate and it's telling me this is arthritis grade 2. Instead of having that, you do all of the heavy lifting using your convolutional neural network, which is basically a deep learning model, to predict intermediate things or concepts that make sense, that a human observer would understand. And then to use a very simple linear model or any other variant that is very simple to map these concepts to the final task you're trying to predict. So the advantage of doing this kind of thing is that the deep learning, which is known to be fantastic functional approximators, these models do all of the heavy lifting, but then at the final step, you have concepts that someone can go back to and understand why this model predicted this is grade 2, for example, because there are bone spurs. And I'm not a radiologist, but you understand what I'm trying to say. So this one advantage of of concept bottlenecks. The other advantage is that I talked about the scale of the whole slide. Having these kinds of bottlenecks, having these intermediate entities as opposed to images or patches can really allow you to fit all of the data from one slide into one model, as opposed to patches, because if each image contains, even a small patch size contains, thousands of pixels, whereas the number of nuclei, even though it's a lot, it's still much smaller than the number of individual pixels. So it has a modeling advantage too for people who work kind of in the field. So going back to what we're trying to do, really the tumor microenvironment is composed of two things: tissue regions and cell nuclei. Tissue regions are these things that are kind of broad patterns that you can see at a low power or low magnification. And here we're concerned with maybe 4 key region types. Denoted here in red is the tumor. The stroma is transparent, so all of this pinkish material is the stroma. And then the bluish stuff is your immune system that is fighting the tumor infiltrating lymphocytes, which are known to correlate nicely with survival. And finally, there's necrotic tissue in yellow, which is dead stuff, dead tissue. So all of this is done at a kind of lower magnification. And then we have the cell nuclei, which is the focus of this work, which are these very detailed elements that really comprise a lot of the difficulty and probably a lot of the interesting stuff in your slides. So you really need both to understand what is going on in the tumor microenvironment, which of course is important when you're trying to understand what is happening to the patient, and at a, to really map it to what's going on from a slide to a patient. So in this space, we try to address the small unlabeled data, big labeled data issue that I just talked about. And we try to address it using open data, basically. So open data is the idea that we try to publish a lot of our annotations. And this is really facilitated by platforms like grandchallenge.org from our friends in the Netherlands, collaborating group there. And basically, and of course, it's facilitated by the fact that the underlying slides themselves are in the public domain. So there's no worries regarding to HIPAA regulations or things like that. I would imagine making our data public maybe a little bit more difficult in other situations. So in any case, we took these TCGA slides from a particularly aggressive type of breast cancer known as triple negative breast cancer. And then we tried to crowdsource the annotation of these structures, the markup, the delineation of these structures. So in 2019, we published about 20,000 region annotations. They're generated by non-pathologists and then validated and corrected by pathologists. So basically, a lot of the heavy lifting in terms of time commitment is done by non-pathologists. But of course, to ensure quality, pathologists look over the work and make sure it's accurate. And then later on, we took this to the kind of higher magnification or next level, and that's our current preprint, the work we call NuCLS. Both of them are available publicly for use by researchers, and I was very happy when people contacted us for even commercial use and things like that. It's very nice to see this field already making use of this work. So why do we need this kind of thing? I'll quickly, I won't spend time on this, but basically, when you have all of these elements, you could do things like pathomic genomic correlation. So you can ask, what are the characteristics of tumors where a particular gene is highly expressed, or a particular patient subpopulation are known to have worse survival outcomes? And this is one spin-off from our 2019 paper, where we, our collaborators, our chemistry collaborators showed that high expression of a particular transcription factor known as MPK3 is actually a transcription regulator, is associated with worse clinical outcomes in African American breast cancer patients. And our region data was used to kind of provide further validation of this, showing that people with the more aggressive tumors have kind of more infiltrative tumor patterns when you measure them using our computational approaches. Of course, this wouldn't be possible if we don't have the data to map all of these structural elements in the slide. Another sample clinical application is scoring the tumor infiltrating lymphocytes, as I mentioned, or TILs. As I mentioned earlier, these are becoming extremely important, and there's a lot of hype around it, justifiably so, because they're correlating very nicely with survival. So if you see more of these, it means your body is fighting back the tumor, which is a good thing. So pathologists are trying to come up with objective ways of quantifying these things. And, led by Dr. Roberto Salgado, we published this report, basically talking about the various things to consider when developing computational algorithms for scoring TILs. And it's really not as simple as just detecting all of the lymphocytes in any area and looking at the global scores. I mean, it could be correlated with survival, but if one were to capture the clinical guidelines, what the clinicians do in real practice, they typically would do the assessment in a focused region, avoid certain areas, and if one were to really try to translate all of the clinical trials that have been done, or at least the prospective studies with the clinical scoring, you want to be close at least to the clinical scoring method, which would require some form of mapping at the level of regions as well as the level of nuclei. One of our algorithms that we did for TIL scoring using these annotations basically showed that the computational TIL scoring using computational mapping stratifies patients in terms of survival outcomes similarly to a pathologist. And when we look at the agreement of algorithms trained using our data with the pathologist scores, basically what you're seeing in this plot in the top left is pathologist 1 versus pathologist 2 scoring. Each dot is a single slide. And then when you see in the panel D here, where my mouse is, this is our algorithm versus the consensus of the pathologist. So what they both have agreed is the score. So in the first panel on the top left is they're scoring things independently, and you can see that there's some sort of variability between them, of course. While you can see in the right here is they sit down together, they give a score, and now we compare that score to our algorithm. And you can see that the correlation coefficient is strong and similar. It's actually a little bit higher than the pathologists have with each other. And what you're seeing on the right here is just some of our existing work preparing kind of better and better algorithms for quantifying these structures, these tumor infiltrating lymphocytes, using the annotations that we've obtained. And that's part of a TIL scoring global effort that Roberto Salgado and colleagues have championed, and we're one of the groups that are participating. It's by invitation, but I assume if someone wants to contribute their algorithm, they can directly talk to Dr. Roberto, and he would probably welcome that. Basically, they have a big set of slides that they've collected from multiple institutions in different places around the world, and corresponding scores from pathologists, including Dr. Roberto himself, but others. I'm not sure how many, but I think it's 10 plus pathologists per slide, if I'm not mistaken. And what they want to do is they want to have this big reader study where they just compare various groups, algorithms to each other, and see basically just the correlation between them, and can computational scoring really match the manual scoring. And of course, if it does, that would be good, because computational scoring is more objective and it could speed up the workflow. And Dr. Roberto can provide more details, but that's kind of the extent of my knowledge of the task. I know Brandon would like to know more about this task, but as far as I'm concerned, it's basically something that is hosted on grandchallenge.org, the same platform where we published our data sets, but it's private, and it's only by invitation only. Even I don't have the data yet. We have some sample slides until we are ready to submit. And basically it's going to be a big reader study, international study, comparing all of these computational models. But again, it's all about data. So you cannot measure these things unless you have data, and that's what this work is focusing on. Okay, so I think now I'm ready to really move to the second part of the talk, and I would like to solicit questions about the first part first.
Hi, Mohamed. I'm going to take this quiet moment while other people find the mute button to say that, you talked about platforms and you just only moderately mentioned TCGA, but I want everyone on the line here to really recognize that that's an NIH initiative that stands for the Cancer Genome Atlas. There's also the Cancer Imaging Archive, and more recently, an Imaging Data Commons, and all these are resources for sharing image data and also the patient information along with it, and then also, like you're hearing Mohamed, the interpretations of clinicians. So make sure you give that a little more play, because for one thing, I know your group is funded by them, and I think it's also a really excellent resource out there. And regarding Roberto's projects, he couldn't make it today. Something came up in his family, but I really am hoping to get kind of a teaser timeline of his machine learning project, so that everyone can see the magnanimity, is that the right word? The largeness of the data that he has, not just in cases, which is large from several clinical trials with, that's probably tracking a patient over time, but also in terms of the number of clinicians that are annotating these images. So, and then this project obviously also includes some real experts in computational pathology, and really the abilities and the tools to access those images are something to really capitalize on and internally increase our expertise with. And Mohamed, that I'll be contacting you more as I try and take advantage of this more with some of my colleagues.
Sure.
Any questions? All right, I'm gonna watch the mutes. I will mute people as we go, just to give Mohamed a clean slate to speak.
Okay. Should I go on then?
I think we're ready. Yeah, I think we're ready. Okay.
So here is, I'm gonna quickly go over the details, because it's kind of a public talk, but essentially, we're crowdsourcing, right? So we're collecting data from non-experts, which means that you have to be really careful about what data you're collecting and how much you trust what you're collecting. By the way, the non-pathologists here are medical students and fresh graduates, which means that they, or graduates, not necessarily fresh, but which means that they have some exposure to pathology. So they have some expertise, but typically in medical school, one doesn't really have any deep exposure to pathology, only superficially. But they tend to have, their incentives tend to be more aligned with doing annotation work. They need the research, they have more time, and whatever the reason is, their incentives, they're more incentivized to do the work. There's also more of them. So we relied on that fact for them to do the heavy lifting, and then for pathologists to really correct all the data. And as you can see here, most of the nuclei that were annotated were in 3 big classes: tumor, fibroblast, and lymphocyte. And then, of course, there were some, small, numbers of classes here and there with variable levels of trustworthiness of the underlying data. One of the things that we decided to do, and it reflected actually in our accuracy measures, is whatever people annotated, we decided to kind of group things based on clinical reasoning. And this is also what is done in a lot of the kind of the machine learning and object detection studies, is that they group classes into classes like the main class set, as well as super classes, which are kind of what you really care about, what you really cannot get wrong. And we also did that just to see the utility of our crowdsourcing approach. So the way people collected data is we showed them suggestions. We have a platform called the Digital Slide Archive. And basically, we had this weak algorithm that generates nucleus suggestions, right? We call them bootstrapped in the sense that it's self-starting. No, we don't have any annotations to produce these. These are produced using things that don't require any data to produce. So we give them some suggestions, and the suggestions are both at the level of the nuclear boundaries, but also they have region suggestions from the previous study that we created. So they know what the label of the region is. For example, this is a stromal region. So they know this is a stromal region, and they see a bunch of suggestions from a weak computational algorithm. And we tell them, if you see something you like, if you see a nucleus you like, place a dot inside it. And if you don't, just place a box around it. And by the way, whether they place a dot or a box, they always choose the class. So even though they see a suggestion for a particular class, that shouldn't, they still need to choose the class actively, provide a specific class for the nucleus. For example, lymphocytes are shown in blue here. And then we ingest this data, these annotations, into a database, and match the points that have been put with the polygons that have been suggested, and then we create this kind of cleaned up data, which is really a hybrid of segmentation boundaries and bounding boxes. And we really haven't seen this kind of work before, even in the non-medical literature. But we argue, at least, that it's better to handle having this kind of mixed data than to really ask people to go ahead and draw boundaries manually, because that's a very time-consuming process, and it's actually one of the limitations, one of the reasons this field has been kind of slow to generate data of a large scale. We argue that you should handle this kind of data algorithmically, as opposed to ask people to just invest more time. So the way the suggestions were generated, I'm not going to go through the details, but we used a model called mask Rcnn. So we initially had some very weak algorithms that rely on existing image processing methods that gives very noisy nuclear boundaries. I'm showing a particularly noisy image here just to illustrate the concept. And then we inherit the labels from the regions. So we say if any nucleus in a lymphocyte-rich region, initially, let's consider it a lymphocyte. Any nucleus in a tumor-rich region, let's consider it a tumor, right? So that gives us an initial data set that we do a functional approximation on top of, using a model called mask Rcnn, which is an object detection model that people use for detecting, I don't know, buildings and cats and dogs and so on. And essentially what this step does for our machine learning folks is it's a function approximator. So there's no training and testing separation here. It's just tries to find, it's like fitting a regression line through noisy data, right? So this gives us the suggestions that then the participants look at. These are, the boundary and high power suggestions. And then the participants can look at both the high boundary suggestions as well as the region suggestions when they make their final assessment of what that nucleus is. And that's how we generate our data sets. Now, we generate two types of data sets. We have a single rater data set and a multi-rater data set. And actually, it's data sets with an S because there's a bunch of them. So these serve different purposes, although one can imagine if someone is replicating this effort in different scenarios, they may choose to use one of these or the other. But for a more complete study, we generated both. So a single rater data set, as the name implies, is where each field of view is marked by only one participant. Of course, one participant can mark multiple fields of view, as you can see this guy here or this gal looking at the top two slides. But each field of view just gets one person. And then they're free to ask questions on the Slack workspace, which is where a lot of the communication was happening. And then the pathologists would respond, say, yeah, that's a plasma cell, it's not tumor, answer questions. Even sometimes they would post links to the slide and the pathologist would go and provide general advice. And then once they generate this data, there's a couple of study coordinators who kind of homogenize everything, make sure everything is standard, and also reply to the final pathologist report. So the pathologist is shown this high-level report of each field. They see the field of view with, let's say, 30 or 40 or 100 nuclei, and they give an assessment whether, yes, overall that's good, that's okay, but needs certain changes, and they say what those changes are, or it's bad. And then the coordinators would go ahead and make these corrections. So that's the single rater data set. We use that for most of our data collection, and the reasoning here is we would like to maximize the number of ROIs that we achieve. Now, the other approach that a lot of groups take, and of course it's also a good approach, is using multi-rater data, which is to have different people annotate the exact same field of view, and then use them to compare the people to each other and to infer what the real labels are. So there's two purposes here. We're comparing people, seeing which where are the outliers from the non-pathologist, or even from the pathologist, who are the outliers, and make sure we account for that. But also to infer, because we have many annotations from different people, one singular label from everything. And when we infer that from the pathologist, we call it the p-truth, because it's pathologist truth. And if we infer it only using the annotations from the non-pathologists, we call it the inferred NP labels, because it's not truth. They are non-pathologists, so they're maybe there is a wisdom of the crowd, but it's not really truth as you would get from a pathologist. We have, in this particular study, we have a couple of experimental controls just to make sure we're measuring the right things and getting good metrics. So our main data set, our main multi-rater data set, is called the evaluation data set. We also have a control called the bootstrap control, where they look at the this data before the refinement by the function approximator. So you'd expect this to be slightly worse than the refined data. And we have the unbiased control, where basically we just show people the field of view without any suggestions, and we tell them to annotate. And this is, it just measures self-consistency with or without the suggestions. So,
Mohamed, can I ask a question?
Yeah, go ahead.
So you, in a multi-rater situation, it's not clear to me, do they do the annotation independently and then you combine them through some algorithm, or
Yes.
they work together in the annotation? That part I didn't hear clearly.
Yes, that's a good question. So the multi-rater data is completely independently. The same person would annotate under different conditions, of course. So the same person would do unbiased, and then there's a, wait a little bit and then do the bootstrap and then do the evaluation for the same field. But different people, they're completely independent. That's for the evaluation for the multi-rater. But for the single rater, there is this collaborative effort. So these are two competing approaches to getting data, and we sort of just use both and analyze both.
So may I ask the infer the p-truth or NP labels, what sort of algorithm? I remember you mentioned in the paper with the EM algorithm to infer that truth from multiple readers. Is that right?
Yes, that's correct. That's actually this slide. So before we do the EM, we first have to map people to the same frame of reference, right? Because they're placing these in different physical locations. They're detection data. So we have to do some sort of clustering, which is what we do. We do an agglomerative hierarchical clustering of the data. So, these bounding boxes, we convert this into an embedding space where the distances between the points are the amount of overlap between the boxes. And then we do some clustering, right? So basically boxes that are closer or that are highly overlapping with each other would map to the same cluster, indicating that this is one nucleus. So the end result would look like, okay, sorry, I'm going to stop moving my mouse. So the end result would look like this, the bottom right of panel A, which is basically you have unified locations, right? And we call them anchors, like potential locations for nuclei, and the matched labels from the participants, including a quote unquote undetected label, right? This person just didn't place a label here. This step is nice because it facilitates doing label inference. Now we have a single frame of reference and we can use expectation maximization, which it basically just uses all of this data to get some label, like some aggregate label. Let's say we do the averaging over everything for the sake of argument, and you get some initial label for each nucleus. And then using that label, you estimate the reliability of each participant with respect to each class. The reason you can do this is because now you can look at outliers, right? You have a distribution, and let's say if 20 people call this lymphocyte and only one person called it, I don't know, tumor, then chances are this person is wrong. So you use that information to further refine your initial label, which was probably bad because it's random. Ish, and then you do this cycle multiple times to end up with the best estimate, your best estimate of what the real label of the nucleus is. So I think, are there questions, or do I move on to the next part of this? So this is the methods, actually.
This is Wei again. I have a question. Have you considered or thought about the other methods in combining multiple readers segmentation? For example, staple was a old method now, but have you considered other methods, or what's your motivation of doing this new method here?
Right. So are you talking about the second part, the expectation maximization, or the clustering component?
Well, I view that both steps are your approach of combining, right? Combining multiple readers, defining a truth, right?
Right. So there's a study that I can probably share with you offline that looked at, it wasn't looking at detection only, it was just looking at multi-rater data in crowdsourcing experiments. And it found that they were comparing things head to head, and they found that the only real problem happens when you do average, majority voting. That's when you get a real problem. Other than that, they didn't actually find any systematic difference between things like expectation maximization and Gaussian processes and a lot of these other techniques for inferring truth. And they actually found that expectation maximization, even though it's fairly old, that is actually it's a quite a robust method. I can share with you that work offline.
So, Wei, we have a separate paper that looks at how to do this with Gaussian processes, and it's being reviewed. So once through the pipeline, we can share that with you too.
Great, thank you. Yeah, I'm interested in looking at that.
Yeah.
Yeah, and if I could, just to make sure I understand, really the staple method is your panel B, because in the staple method, there's only one object being segmented, and so there's no issue with trying to cluster things and say that they're segmenting the same thing. And so that's that panel A, is trying to get down to individual objects. And then
Yes.
panel B is to basically, and it still sounds actually a lot like the staple method, but it's the staple method on each one of those objects, or maybe all of them at the same time.
No, you're right. It's per object. Yeah.
So your picture kind of shows it all happening at once. You don't build.
Oh, you're saying here. Yeah. This is done globally. So we look at all observers, all objects, and we estimate their reliability.
Whereas staple really focuses on one object.
Ah, okay.
I believe. Isn't that true, Weiji?
Yes, I think so too.
That's a nice generalization, of course.
I mean, it's always nice to see how these things compare. We have the data published, including the individual multi-rater data. It would be nice to see. It doesn't hurt, you know.
Yeah, I mean, it comes from need, right? Here you've got all these different targets. In radiology, you had one lesion. And,
right.
So great, thank you.
Mm-hmm. So I'm not going to spend time on this, but basically this is just showing that the accuracy is high. So what you're seeing here is the detection accuracy of the inferred label from the non-pathologist. It achieves an average precision of 0.68, which is fairly high actually when you compare to known data sets like the mask Rcnn COCO data sets. Classification accuracy, again, is fairly high when you look at both classes and superclasses, but it becomes lower when you do macro averaging. So when you take these uncommon classes into consideration, the accuracy lower. So that's just one thing to keep in mind. Another thing we did is we did some simulations, and essentially we're seeing how many people per field of view do we need to classify a particular class accurately. Now, this is fairly specific to our problem, but it just illustrates that this is class dependent. So for classifying stromal cells accurately, you actually need 6 or more people per field of view in the multi-rater data. Whereas to classify tumor or lymphocytes is an easier problem. 2 people are actually enough to get that. But of course, the more the better. Another thing we show.
I'm sorry, can I interrupt a little bit here? I think there are a lot of information here. I'm not clear. Maybe I'm trying to understand some basic things. Like if you look at the B, the RC curve. So what's this about? This is about what's the decision variable and what's the ground truth here? What are you trying to show here? Can you spend a little bit more time on this?
Yes, sure. Sorry, I glossed over it. I'm sorry. So the truth is our inferred truth from the pathologists, right? Okay. And what's being compared here is, first of all, everyone individually. These are scattered. The non-pathologists are these circles, and the pathologists are the rectangles and squares, triangles. And so that's the truth is the inferred truth from pathologists, and these are the individual people. Now we want to see how the non-pathologists are collectively doing. So we now compare our inferred labels, right, from the non-pathologists to the truth from the pathologists. And that's what we're seeing here, this ROC curve.
So what's the unit of analysis here? Is it pixel-based? You're looking at it based on the segmentation truth, you're looking at what's the true positive, false positive here?
Right. So this is classification accuracy. Everything is at the level of individual objects, individual nuclei.
Okay. All right. So this is for nuclei. Okay.
Yes. Everything actually in this analysis.
You have multiple types of objects in your segmentation. So this is about the classification for nuclei.
Yes, nuclear classification. That is correct.
And how many? What's the sample size here? roughly?
Here, this is our multi-rater data. So 1,400 unique nuclei. Of course, in terms of number of annotations, that's a lot. It's I think around 100,000. But unique nuclei after clustering, that's about 1,400 or so for this multi-rater data. Of course, the single rater is much larger.
Okay.
Yeah.
May I ask a question? Sorry.
Yeah.
About the lower right panel. I think how many NPs should annotate? Can you talk a little bit more about? I guess the horizontal axis is the number of NPs per FOV, and the vertical axis, it's the AUC per reader again?
No, this is the AUC under that curve from panel B, right? And of course, we randomize, we repeat it multiple times, so there is a distribution around it, right? So this comes back to the simulation methodology. So we have multiple people. Our actual data is you have, let's, for the sake of argument, for the sake of this illustration, let's say we have 3 FOVs and let's say 10 people, right? Our actual data has 7 people per FOV, or 10 people, all 10 people annotated all the FOVs. For our simulation, we decimate, we delete data, and we see what the accuracy of our inferred labels from these people. Obviously, now you have, instead of 6 people annotating the same thing, you only have 3. So you'd imagine your accuracy would be lower, right? But here we're showing that, yes, the accuracy, of course, is lower. The fewer people are annotating the same image, but this is dependent on how difficult the problem is. For detection of stromal nuclei, for classification of stromal nuclei, that's when you need more people.
Yeah, and I just wanted to emphasize that that's really something that should be paid more attention to, because there is, these ideas that, regardless of the task, you can use the same number of truthers. And I think this figure very nicely shows that it really depends on the task. For some tasks, you really need to have to go to a large number of truthers to get something close to what can be used, let's say, to make a regulatory decision about a system.
Yes, yes. I think that's precisely what this is trying to say. And yeah, absolutely.
Hey, Mohamed,
Yeah.
you said you were going to gloss over that slide, but you can't present results data to this crowd without getting, it's go big or go home. If you don't want this discussion on that slide, let's not get it. So I see.
I think what I will gloss over is actually the modeling, because I think it's less
definitely
relevant to the data. Yeah, it's less relevant to the data and more relevant to what we specifically did. So I think I will leave this as the last slide.
Because I have I think we should have the discussion that these 2 slides probably deserve. Yes. And I think you're right. Modeling, we can all work on algorithms at another time, but this is at the FDA, it's all about performance evaluation and about understanding that critical question of how many pathologists you need to get an accurate, valid truth,
Yeah. Absolutely. So even like here, so maybe this is the last thing I'll discuss, and then we can go back to the truthing simulation. Here is just showing one way in which we can have controls. So, as I discussed, we have an unbiased control. We don't show any suggestions. We show weak suggestions, and then we show our actual, strong suggestions. And we see how many clicks how many nuclei do people think are accurate. And this is actually looking at annotations from a single person, the same person actually, who happens to be a junior pathologist here. And you can see that, as the quality of the suggestions improves, people click more nuclei. And more than that, the nuclei they click are also more accurate. So basically, this is just validating the idea of how we generated these algorithmic suggestions. And we also look at the self-agreement. If the same person in this panel D here, the same person makes the same prediction whether or not they're seeing a suggestion, and we measure this using Cohen's kappa, of course. And you can see that they have a substantial agreement, but also that it improves as you group things together. So with that, I'll open it up for discussion. But before that, I just want to thank everyone who contributed to this data set, and of course acknowledge the funding that we had. And yeah, I'll open it up for questions, especially regulatory ones, of course.
Can you go back to the last slide? You showed that with unbiased control or suggestion control.
Sure.
So I think I'm trying to understand this. I think maybe there's different terminology, because we used to talk about manual segmentation, semi-automatic segmentation, or fully automatic segmentation by the algorithm. So the manual segmentation without any algorithm that's the unbiased control here. Is that right?
That's correct. Except here we have a hybrid data set, so it's not really segmentation. It's a mixture of bounding boxes and segmentation. Yes, you're right. So the evaluation is the semi-automatic.
And it's not totally manual because you're already putting red bounding boxes around things.
You mean for the unbiased or for the evaluation?
The unbiased.
Well, for the unbiased, this is what the person placed actually.
Okay.
They placed the boxes.
All right, thank you.
Yeah, for the unbiased control, my understanding is it's manual annotation by themselves, and the other two you basically give them software assistance. I think sometimes it's called semi-automatic annotation.
Yeah, I would say add one more row here showing what the starting point was.
Yes, so this, you're right. We show this in just a sec here. So the unbiased, that's the starting point, right? That's the bottom. For the evaluation.
I don't get it there. I don't understand, because you're already saying suggestions. So to me, a suggestion is not fully manual.
Oh no, like for the unbiased, there's no suggestion. I'm just showing that there's nothing.
Yeah, I think that's a good point, Mohamed. Maybe we should, confer on that before we resubmit.
Yeah. And on the last slide we were just on, just show what the user sees when they first turn to that region of interest.
Sure.
That will help. That will help me at least.
Sure, yeah. We can add like a fourth one here, like what they see and then what they added.
Mm-hmm.
So I was looking for some. You have a lot of results. You did a lot of work. So I was looking for some specific things here. Is that in the beginning, I remember you showed that there's substantial variability among the pathologists themselves. So I'm wondering, do you look at like the agreement among the junior pathologist or non-pathologist versus the senior pathologist in using these suggestions or software assistance, how that will reduce their variability, for example, or affect their performance even. So my hypothesis is that the non-pathologist may rely on the software, and therefore you may be able to see a great reduction of variability by using the software as assistance, whereas the experts may choose to ignore. I don't know, but I'm wondering about that kind of results. Do you have that?
That's a good point. So we didn't see the inter-rater variability. Actually, we did, but we didn't compare it head to head. I can look for that data afterwards. What we did compare is the same person compared to themselves when they're not seeing suggestions versus when they're seeing suggestions. And you see the scatter here, the squares are the junior pathologists or the residents, or at least people who didn't have 2 years of training, and the rectangle and the triangle, are the senior pathologists. And you can see that the senior pathologists have higher agreement with themselves, whether or not they see suggestions. In general, you see this, not in general, but you see it at least when you look at the fine-grained classes, not when you group things. What you see very clearly, though, and it's a consistent pattern, is that the non-pathologists are kind of more impressionable. They see a suggestion and they would change what they would call a nucleus depending what the computer suggests. But what we show in panel E here is that even though they're more impressionable, this actually results in an improvement in their accuracy. So they're impressionable in a positive direction. They were thinking of the wrong label, and then the computer suggests something, and they're like, oh yeah, that looks like it. And that's why you see the area under curve for when they see suggestions, which are these top two curves, is higher than when they don't see any suggestions at all.
Of course, that's driven by how good the suggestions are.
Absolutely. Absolutely. It's highly algorithm dependent, and but I would argue that like our initial algorithms that we use are actually pretty weak. They're like just Gaussian and thresholding and things like that. Like, let me show you this slide here. Like these algorithms are, we use, you know, all of the things that people use, and I forget the watershed and Gaussian and Otsu thresholding and so on. But really, it was to showcase the potential of this approach. We really use a weak algorithm here, and the mask Rcnn is doing a lot of the heavy lifting in terms of detection-wise, improving the detections. Maybe segmentation and maybe the classification, not so much, but yeah, it is algorithm-driven for sure.
Okay. As we're getting close to the top of the hour, I wouldn't mind revisiting the slide with the performance results again. And anybody that isn't getting their questions in now, feel free to try and log them in to me by email or something, and I'll try and pass them on to Mohamed. But certainly, if you're going to be sharing the reader data here, there's a lot of great statistical method, not just applications, but method development that this data is just primed and ready to support. I mean, even you use the word simulation in that figure, but the way you described it is not a simulation. It's not modeling and rolling the dice to come up with performance results. You are starting with 32 readers and then you know, eliminating or selecting 2 of them to see, and very randomly selecting 2 of them to see that performance. So when we think of simulation, we really think starting with models and whether they're physics or statistical type models and rolling the dice at each step to create that data. But you have the ability to quote unquote simulate because you have so many readers.
Absolutely.
And I think that's a huge resource to take advantage of.
Absolutely. Feel free. You know, I was, while you were talking, I was showing, you know, it's very easy. Like it's literally, you just click download links and you can get individual raw data, inferred labels that we have, and you know, this is in the public domain for anyone to use and to test their hypotheses on. That's why it's open source.
And is this the launching point for this web page in the archive article, or is that another link that I have not yet gotten?
It's in the archive article, yes.
Okay,
it's
so
just
like
scicegroup.com/nucleus.
This is the supplementary materials that you created outside of the journal publication.
No, it's actually in the apps, in the like if you go to the paper itself, like literally this URL in the abstract.
Right, but it's not, the journal is not hosting the supplementary materials. You are creating these and hosting them yourself.
Oh, yes, that's correct.
Do you share the data? Do you share the code also? I don't see that in the paper.
That was my next question.
The code is there too.
Okay.
It's there in the paper somewhere. You know, it's like at the end. Yeah, at the end there is. Yeah.
Great.
Excellent.
We're still cleaning it up, but it's there for initial assessment.
Great, because especially that clustering method and the EM machinery is probably really useful in other situations. Any other questions as we wrap up? We're at the top of the hour.
Yeah, it's 3:01. If you have any other follow-up questions, you can also send an email, or this chat also is also available after the meeting, actually. The chat channel.
I don't know if we're going to monitor it directly, but definitely hit me or Weiji with an email and we'll connect with Mohamed. Mohamed, this is great work. Gosh, huge amounts of data. What a resource for the community. Appreciate you speaking about it today. And I look forward, and I hope that the paper gets accepted quickly, and you get to put that on your CV along with the other great stuff you've produced.
Thank you so much, Brandon and Weiji, for the wonderful introduction, really great discussion, and everything. And I really look forward to continuing to work with you.
Absolutely.
Yeah, really, thank you very much for the great presentation. All right, with that, we can close out the meeting. Thank you, everyone, for attending.
Thank you.
Bye-bye.
Bye-bye.